Today we're going to look at Chapter 13 over the peripheral nervous system. Why is the peripheral nervous system even important? Well, it does contain sensory neurons and motor neurons that carry out two of the three functions of the nervous system. So it can carry sensory information to the central nervous system and motor information from the central nervous system. So it not only contains the neurons that carry sensory input to the brain and spinal cord, but it also contains nerves that carry orders sent from the central nervous system to voluntary muscles and other effectors of the body. So it allows us to move, take care of our own needs. The effectors of the body include muscles and glands, so we can see muscle contraction or gland secretion. It's able to provide links from and to the world outside our bodies, and anatomically they are ghostly white nerves, or what is the peripheral nervous system, consists of ghostly white nerves that thread through virtually every part of the body, and it does enable the central nervous system to get and send out information. So here we can see in this image the vagus nerve. They are along the carotid arteries, and they're kind of contained therein and following the same route. They're ghostly white and thin and kind of very fragile structures. Here is a review slide. So this is figure 11.1. And we're seeing all three functions of the nervous system: sensory input, integration, and motor output. We've talked in depth about how the central nervous system, both the brain and spinal cord, function in integration, or processing and interpretation of sensory input. But it's the sensory input and motor output that are the functions of the peripheral nervous system. A sensory input is carried along a sensory neuron to the central nervous system, and a motor output is carried along a motor neuron to effector organs. Again, effector organs include muscles and glands that will either contract or secrete. The peripheral nervous system includes everything outside the central nervous system. So anything that's not the brain or spinal cord but is nervous tissue is a part of the peripheral nervous system. The peripheral nervous system, again, is all neural structures outside the brain and spinal cord. So things that would be included would be sensory receptors, peripheral nerves, the associated ganglia to those nerves, ganglia are cell bodies, and efferent motor endings. An example of an efferent motor ending would be the axon terminals of a somatic motor neuron near the sarcolemma or junctional folds of skeletal muscle. Again, the peripheral nervous system is going to provide links to and from the external environment. Here we can see the two parts of our nervous system, the central nervous system and the peripheral nervous system. So you can see that the peripheral nervous system is actually the larger part of the nervous system, even though integration is a very important function for the nervous system, and that's carried out by the central nervous system. We can see that the sensory or afferent division is sending information to the central nervous system, and that motor output signals, or the motor efferent division, is carrying information away from the central nervous system. We do use the central nervous system to survive because we need sensation and perception in order to process our environment. So even though the central nervous system is processing that information, we're getting all of that information and sending it out into our peripheral nervous system. So sensation is the awareness of changes in the internal and external environment, and perception is the conscious interpretation of those stimuli. We need both in order to survive and interpret our environment, and raw data is not enough in order to make decisions. So not only localized deep pressure, but the awareness of discomfort and if there was a pebble in your shoe are both required in order to have a response. Perception especially, because perception determines response. So sensation is the raw info, and perception is memory and previous associations with a stimulus, whether you like something or don't, and that perception will create the motor output, the effect on the effector organs. So you can sense information without becoming consciously aware of it, but then you don't truly perceive it. Next, we're going to look at the general organization of the somatosensory system. Somatosensory system is the part of the sensory system serving the body wall and limbs. It receives input from different types of receptors, including exteroreceptors, proprioceptors, and interoreceptors. We're going to see that these types of receptors are able to transmit information to the central nervous system about several different types of sensory modalities or types of sensation. So if we talk about what does somatosensory mean, soma means body, so somato is just a longer root word for body, and then sensory is talking about sensory stimuli that would travel to the central nervous system. So we're going to look at basically sensory information about the body being picked up on or upon by certain types of receptors. Again, those receptors are exteroreceptors, which are sensory receptors responding to stimuli arising outside the body. So auditory information or visual information would be picked up by exteroreceptors. Proprioceptors are receptors that detect motion or position of the body, so they tend to be positioned around muscles or joints or near tendons, or even in the inner ear, which has to do a lot with equilibrium. And then there's interoreceptors, which are sensory nerve receptors responding to stimuli within the body. Think visceroreceptors as well. Think your organs, whether or not you have a full stomach or a stomach ache, or if you can tell that food's being processed through your small intestine because maybe a pressure in your lower abdomen. Those would be signals picked up by interoreceptors. Next, we want to talk about 3 layers, or we can call them levels, of sensory integration. So there are levels of neural integration in sensory systems, and we'll start with the receptor. There is a receptor that has to pick up on a sensory stimulus. Then once that sensory stimulus travels in an axon, we've reached the circuit level, where we're processing signals and getting them towards the central nervous system. Remember, in ascending pathways, there are 3 neurons in the circuit, a first, second, and third order neuron. Then the perceptual level, where the information has reached the brain and is processed in cortical sensory areas, or the cerebral cortex. So sensory information is generally going to be relayed toward the head. Sensory information is also afferent information, but it's going towards the central nervous system. It can be processed along the way. There are different parts of the spinal column that function in reflexes, and sometimes information doesn't even have to reach the brain in order for us to respond. So let's look more at the receptor level. For a sensation to occur, a stimulus must excite a receptor and action potentials must reach the central nervous system. So a graded potential must be made. That graded potential must be strong enough to incite an action potential, and that action potential has to travel along a couple of neurons to reach the brain or spinal cord. So in order for this to happen, the stimulus energy must match the receptor. So we're not going to apply different chemical components to a mechanoreceptor because a chemoreceptor and mechanoreceptor do not take the same type of input. So a touch receptor would need something like mechanical pressure, stretch, or vibration that distorts the cell membrane of that receptor. The stimulus energy must also be applied within the area where the receptor is located, or that receptor's receptive field. And that's just going to be the area that the receptor monitors. And then the stimulus energy must be converted into a graded potential. Remember that graded potentials initiate action potentials, and that initiation process is called transduction. So we have 2 types of receptors. We have our special sense receptors and our general sense receptors. General sense receptors usually use modified dendritic endings in order to transmit a sensory input through or to a neuron. So we could say that the first order neuron and the receptor are almost the same thing. We're just looking at modified dendritic endings. So we're not going to have the signal have to jump from receptor to first order neuron and then up the pathway towards the brain. So in general sense receptors, think mechanoreceptor, for example. We'll see that the graded potential is going to be called a generator potential, and that's only because it can generate an action potential if strong enough. We're just calling it something different instead of a graded potential because it is occurring in a modified dendritic ending. Whereas in the special sense organs, the graded potential actually occurs first in a receptor. And that receptor has to send that signal to a neuron, and the neuron has to generate its very own graded potential. So we kind of have a 2-step graded potential when we're looking at processing at the receptor level in special sense organs. So for the receptor level processing in a special sense organ, we're going to call the first graded potential a receptor potential. Remember, in the general sense organ, there was only one graded potential that occurred, and it was a generator potential. So here we have the stimulus creating a graded potential in the receptor cell. That's what we're calling a receptor potential. Then that receptor cell, when stimulated, would release neurotransmitters into a synaptic cleft between the receptor cell and the first order neuron. When those neurotransmitters act on the first order neuron, they would generate a graded potential in the sensory neuron, which is the first order neuron, and it would create its very own graded potential again before trying to initiate an action potential. So there's actually 2 graded potentials in this processing if we're looking at special sense organs. But don't forget that for processing at the receptor level, we have to have specificity for the stimulus energy. We have to be acting on the receptor where it can be stimulated, so within its receptive field, and we have to create a graded potential, whether that's a general sense organ, so we're using, for example, a generated potential, or if we need two graded potentials, therefore a receptor potential, then one in the neuron itself. It depends on a couple of different things, and that first and foremost would be if we're looking at a general sense receptor or a special sense receptor. A special sense receptor example would be something like the photoreceptors of your retina or specialized chemoreceptors on your tongue. But a generator potential is for general sense receptors, and a receptor potential is for special sense receptors. But either way, a generator potential or graded potential must reach threshold in order to trigger an action potential. Graded potentials must always reach threshold in order to trigger an action potential. So let's review. We're trying to work through the levels of processing, and we've gone through the first level, which is processing at sensory receptors. So that's the receptor level. Next, we're going to move and look at processing in ascending pathways. So processing from that first order neuron to the second and then to the third order neuron leading us to the cerebral cortex. So that's going to be circuit level processing. And last but not least, we'll look at processing in those cortical sensory areas, and that's going to be our perceptual level processing. And remember, perception will determine a response or the motor output and what effector organ that output goes to. So processing in ascending pathways. Remember, once we reach the circuit level, we've already created an action potential, so that action potential is on its way to the second order neuron. But this is our second level of integration, and the task for the circuit level is just to deliver that impulse to the appropriate region in the cerebral cortex. It really is just a pathway. It's trying to get this signal from point A to point B, point A being the receptor, and then point B being the integration center or the cerebral cortex. So for ascending pathways, we had 3 neurons that conduct sensory impulses towards the central nervous system. There was a first order, second order, and third order sensory neuron. That third order sensory neuron will be able to conduct the impulse from the thalamus, which is the relay station for the brain, to the proper somatosensory cortex. And this will be the last part of processing, and we'll jump from the circuit level to the perceptual level using this third order neuron. Next, we're going to look at adaptation of sensory receptors. So adaptation occurs when sensory receptors are subjected to unchanging stimulus. And we don't see adaptation in all receptors. Some are very fast adapting, and some are tonic or have a sustained response or little or no adaptation. An example of a tonic receptor would be a nociceptor, which is a pain receptor. And it's important that our pain receptors don't get used to pain so that we're able to recognize if something is trying to hurt us, and we're able to protect ourselves against damaging stimuli. But phasic receptors are fast adapting, so we'll get used to these types of stimuli. One example of a phasic receptor would be your olfactory receptors. You can get used to a smell if you're around it enough. So receptors responding to pressure, touch, and smell would adapt a certain way, and we'll get that answer in a minute. Then receptors that respond slowly are things like Merkel's disc, Ruffini's corpuscles, and pteroreceptors, and they're slightly tonic, but they're not 100% non-adapting. But then blank receptors, so the receptors like proprioceptors and nociceptors, are not going to exhibit adaptation. So let's see our answers. So our first answer is receptors responding to pressure, touch, and smell adapt phasically. So they do adapt. You get used to these stimuli. So it makes sense. So pressure and touch, an example for that would be your shirt. You put your shirt on, you may feel it at first at the beginning of the day, but throughout the day, you're not focused solely on every location where your shirt is touching your body, because those touch receptors on your back and your arms, they're going to be phasic. They get used to that pressure, and they stop telling your brain, hey, something's touching me, something's touching me, something's touching me. And that would be annoying if they did so. So it's important that we have phasic receptors, ones that can adapt. And then our next answer is tonic receptors. So if you see anything about pain receptors, know that they are tonic. But proprioceptors are also tonic. because proprioceptors let us know what our body is doing, whether we're sitting or we're standing or we're kind of moving about in our environment, and we want our brain to be aware of what's up, what's down, what are we doing, how are our muscles bent, how is our skeletal muscle responding or contracting. So it's important that both nociceptors and proprioceptors are tonic and do not exhibit any adaptation. Next, let's look at the processing that happens at the perceptual level. So this is occurring in the cerebral cortex. So this is happening after a third-order neuron of the circuit level has reached the cerebral cortex, has been conveyed by the thalamus to the right somatosensory area, and now we're going to process this raw data. We're going to look at 6 major features in order to have proper perceptual sensory information or integration. So first and foremost, perceptual detection, then magnitude estimation, then spatial discrimination, then feature abstraction, next quality discrimination, and then pattern recognition. So a lot of these will make sense to you. So perceptual detection, detecting that a stimulus has occurred. So actually getting the raw data to the right location. The simplest level of perception is perceptual detection. Magnitude estimation has to do with how intense a stimulus is. So were there a lot of active potentials that were sent over a certain period of time? Was the threshold not only met, but beat, meaning that the original graded potential probably was also much stronger. So it's just letting us know kind of how intense is that stimulus that was received. Again, this will be kind of conveyed to the central nervous system via frequency coding, because we never increase the height of an action potential wave. We only increase the frequency of action potentials to let us know how intense a signal is or how strong it is. Then there's spatial discrimination, so just identifying the side of the stimulus. And the thalamus really helps out with that. It's going to send this sensory stimulus to the right somatosensory region of the brain, or the right cortex. So sending that to the right region has a lot to do with spatial discrimination. It lets us know, did the signal come from the elbow or the knee or our stomach? But it lets us know where the site of the stimulus originated. Then there is feature abstraction. This gives us a little bit more information about certain kind of modalities, things like texture or shape. Is it pointy? Is it square? Is it round? Quality discrimination, the ability to identify submodalities of a sensation. So not just texture and shape, but is it sweet or sour, or if it's round, does it have a rough outer edge? Kind of just extra information. So the quality of the texture, quality of the shape. Is it completely round, or are there bumps? Things like that for quality discrimination. And then pattern recognition has to do with whether or not we've recognized this pattern. Is it a face? Have we seen this person before? Is it a song? Do we know the melody even if we can't recognize who sung the song? Things like that. And next we want to move on and look at our sensory receptors more. And we're going to lead up to talking about different types of sensory receptors and not just the 3 broad divisions, which include proprioceptors, interoreceptors, and exteroreceptors. But let's just talk about sensory receptors kind of in general. So sensory receptors are structures specialized to respond to changes in their environment. They will be activated when they receive a sensory stimuli or input, and this will cause a depolarization. Normally a graded potential, if strong enough, will cause an action potential. So the realization or sensation and perception of these stimuli occur in the brain, but the original stimulus will act upon the sensory receptors. So even though the information is going to travel towards the central nervous system because it's sensory, it all begins at the sensory receptor. So let's look at the classification for our different types of sensory receptors. Some we've already looked at, but there's 3 ways to classify sensory receptors: by the type of stimulus they detect, by their body location, and by their structural complexity. So stimulus type. We got mechanoreceptors, thermoreceptors, photoreceptors, chemoreceptors, and nociceptors. Mechanoreceptors respond to mechanical stimuli. A lot of distortion in cell membrane. So they respond to touch, pressure, vibration, stretch, even itch. Anything that would physically kind of distort or stretch or move a cell membrane could be picked up by a mechanoreceptor. There's also, of course, thermoreceptors. Thermoreceptors are sensitive to changes in temperature, so extreme cold or extreme hot would actually be picked up by pain receptors, but as long as it's kind of a general change in temperature, thermoreceptors can pick up on that. Photoreceptors respond to light energy, so we actually find photoreceptors in your retina. Chemoreceptors respond to chemicals, so that would be used for smell, taste, also changes in your blood chemistry. So there's a lot of chemoreceptors that let us know kind of what's going on with your blood gases, things like that. And then last but not least, your nociceptors. Nociceptors are pain receptors, and they're really just sensitive to extremes of any of these other stimuli. Extreme pressure or stretch or itching, extreme temperature changes, super bright light, caustic chemicals. All of those are actually picked up by nociceptors because nociceptors pick up extremes of all these other stimuli. So they're sensitive especially to pain-causing stimuli that are extreme heat or cold, excessive pressure, and inflammatory chemicals. We've already talked briefly about classification by location. Those include exteroreceptors, interoreceptors, and proprioceptors. And I said think visceral organs for interoreceptors because their other name is visceroceptors. So again, exteroreceptors are responding to stimuli outside the body, interoreceptors responding to stimuli inside the body, and proprioceptors responding to stimuli that lets us know what our body's doing. So proprioceptors do inform the brain of one's movements. You can associate exteroreceptors with most special sense organs. So receptors in the skin for touch, pressure, pain, and temperature, but also for the photoreceptors of our retina, or the different receptor mechanical cells of your ear near your cochlea or the semicircular ducts. And then for your proprioceptors, just be aware that they're kind of in regions of your body where information could go to the central nervous system and provide information about what your body's doing. So in joints, in tendons, around muscles, that's where proprioceptors are. Now, depending on the structural complexity, we also can classify receptors. Complex receptors are the ones that are picking up information for special sense organs. And then simple receptors are going to be the ones picking up information for just general senses, not any of our special senses. A lot of mechanoreceptors here. So you can even see for simple receptors, we're going to just have modified dendritic endings. And we've heard that before in this PowerPoint. And modified dendritic endings actually used a generator potential to trigger an action potential. So it's all occurring in that first order neuron. We're going to pick up on tactile sensations or kind of pressure inside muscles. So we see a lot of that for our simple receptors. And then complex receptors, being special sense organs, have to generate a receptor potential in separate cells before communicating with a first order neuron. These complex receptors are going to include receptors for vision, hearing, equilibrium, smell, and taste. So let's look at a couple simple receptors first. There are encapsulated and non-encapsulated receptors for simple. So there's free dendritic endings, not very modified as far as dendritic endings go. They basically are just near areas. They pick up on some kind of nociception or pain signals, but they also chiefly respond to temperature. Then there's Merkel or tactile discs. Those are kind of light touch receptors. Hair follicle receptors detect light bending of hairs. So that's a little bit of pressure, but a mosquito landing on your skin will be a great example of how a hair follicle receptor detects that light bending. So here we can see them. So we have modified dendritic endings. We have a tactile or Merkel disc and hair follicle receptors. So definitely the free nerve endings and the hair follicle receptor are the least complicated. We're just seeing kind of modified nerve endings wrapped around structures or in structures. A lot of these are exteroreceptors you can see and mechanoreceptors. Now, free nerve endings can usually be thermoreceptors or pain receptors as well. If we look over here at the tactile or Merkel disc, we're looking at kind of a modified nerve ending that's more than something that we're used to looking just kind of like a dendrite. Instead, this is kind of a pressure, we'll call it a pressure bulb, for lack of a better term. And it's going to be able to pick up on light pressure as it sits in that basal layer of your epidermis. Then we have encapsulated simple receptors. There's the Meissner's corpuscle, Pacinian corpuscle, and then there's the muscle spindles, Golgi tendon organs, Ruffini's corpuscles, and joint kinesthetic receptors. These down here are listed so close together because they have something to do with either tendons or muscles or something at a joint. So a lot to do with proprioception. So let's look at the tactile or Meissner's corpuscle first. It's an exteroreceptor. And we would find that located in the dermal papillae of hairless skin. So in the nipples, the external genitalia, fingertips, soles of the feet, eyelids. And you're going to see that most of these are going to be exteroreceptors or proprioceptors. One that can be an interoreceptor, though, as well, is a lamellar or Pacinian corpuscle. And it's going to pick up on kind of deep pressure, stretch, vibration. It's very rapidly adapting, and it's in the dermis and hypodermis. So this would be one of the ones that's maybe picking up on kind of the pressure of your shirt on your body. Then the bulbous corpuscle Ruffini ending is going to be looking at, for example, deep pressure and stretch on tendons. And it's also in the dermis, hypodermis, and joint capsules. It's also looking at kind of stretch of collagen fibers. That's what it's doing in the dermis and hypodermis. Muscle spindles, its name says it all. It's going to be modified dendritic endings that spin or wrap around individual muscle cells or interfusal fibers, kind of deep fibers of skeletal muscle. And they're going to pick up on stretch or any changes in length. But we specifically find them around skeletal muscle, particularly in the extremities. And then here is a tendon organ. And we can see a tendon with modified dendritic endings going through it. And if this tendon stretches or bends, that information can be conveyed via a first order neuron, then up the rest of the circuit level to the central nervous system. So this is a proprioceptor. It's using mechanoreceptors to tell information about the tendon and again convey that to the central nervous system. And then joint kinesthetic receptors have kind of a similar appearance, hence them not having their own image. And we're actually going to stop there before we go into special senses. Just go back and review the difference between kind of our general versus special sense receptors as far as graded potentials are concerned. Again, graded potentials using special sensory receptors have to create a receptor potential first and then a new graded potential in the first order neuron before we enter the circuit level. Make sure to review the levels of processing for the somatosensory system. So those include the receptor level, circuit level, and perceptual level. But next time we'll go into the types of special senses and look at their receptors.